The work of Nobel Prize has transformed our understanding of the world. While approaching the Nobel Prize winners in chemistry, we not only learn their achievements, but also the spirit of honesty, tenacity, innovation, and forging ahead.
Hello everyone, welcome to today's class, the Nobel Prize winner in chemistry. 同学们好，欢迎走进今天的课堂，走进化学诺贝尔奖。今天要给大家介绍的科学家是钱永健，把彩色带进生命的科学家。Today I will introduce you a scientist who brings colors into life. His name is Roger Yonchien Tsien. With every new and effective tool, science will take a big step forward, and human society will reach a new peak. The invention of a microscope allowed Leeuwenhoek to observe countless swimming objects in water, and the door to microbial world was opened. The invention of the astronomical telescope also gave Galileo a clear view of the Creator on the surface of the moon, and the movement of sunspots on the sun, giving people a new understanding of the sky. In the late 20th century, green fluorescent protein, shorted for GFP, appeared as a tool that made people seem to have golden eyes. The imaging technology based on the GFP is not only like a light, but also like a microscopy in the cell. It enables scientists to observe the rich and colorful life phenomena at all levels more directly.钱永健is the scientist who has changed the green fluorescent protein, known as GFP, to make its glow stronger, more stable, and more colorful. He is known as the lighting up biology. In this picture, as you can see here, the word Tsien Lab is written by different color GFP. Roger Tsien won the Nobel Prize in Chemistry of 2008 for green fluorescent protein. He was a professor of pharmaceutical chemistry and biochemistry at the University of California, San Diego School of Medicine.
Actually, because of the time, I'll use the version on this computer, I hope. And I think I'm going to use this one because wrong one. It's this one. Go. At least I'll try. Okay, of course, I too must thank the committee of the Royal Academy, the Chemistry Committee of the Royal Academy of Sciences, the Academy itself, and the Nobel Foundation for this tremendous honor bestowed upon me, and for the honor of sharing it with two such esteemed co-laureates. My involvement with the GFP began here with the wish to understand and study cyclic AMP signaling of the most important intracellular messengers inside cells are calcium and cyclic AMP. And I had already done a lot of work on calcium and still have an interest, as you will see, but that did not directly lead to GFP.
Qian's father was a cousin of Tsien Xuesen, who was the founder of the Jet Propulsion Laboratory and early leader of the modern Chinese space program. Roger was born on February 1st, 1952 in New York City. He was a scientist from the early childhood, sketching out chemistry experiments as an 8-year-old in a notebook, which is now kept in the Nobel Museum. His first boy-cow merit badge was in chemistry. In 1968, at the age of 16, he took the first prize in the prestigious Westinghouse Science Talent Search for high school seniors. He attended Harvard College, graduating in chemistry and physics in 1972, then earned his PhD in physiology at the University of Cambridge in England. Tsien had attained a great achievement in his career. He was a member of the Institute of Medicine and American Academy of Arts and Science, the United States National Academy of Sciences, and the Royal Society of London. Besides Nobel Prize, he had also won many outstanding awards. Much of Roger's early work was directly at image-neutral activity, by trying to develop tracers of sodium or calcium ion movements that support brain signaling. By 1980, he had invented Queen 2, a synthetic fluorescent dye that selectively binds to calcium, and had devised a clever way to sneak this dye and other probes into intact cells. This first practical probe for calcium found wide early use in studies of intracellular calcium signaling. As we can see from the picture here, there are several types of calcium ion probes invented by him. Tsien, Shimomura, and Calfee collaborated to discover and develop the green fluorescent protein derived from the jellyfish as a new and soon indispensable research tool. Shimomura identified the crucial jellyfish protein and revealed that it glowed bright green under ultraviolet light. And Calfee showed how it could be used as a biological marker. Tsien found ways to make the GFP glow more brightly and constantly. And he created a full palette of fluorescent proteins that scientists could use to track different cellular processes at the same time. Before the discovery of fluorescent protein, many kinds of organic dyes and minerals that can produce fluorescence has been found. But no matter how much these substances are put into cell, they can only be detected in a constant amount. One of the most important significance of fluorescent protein is to achieve from scratch research in genetic engineering. As long as the target gene carries a fluorescent protein gene, it can be observed in the naked eye whether the product has been inserted into the target gene, as we can see from this picture here. Green fluorescent protein is a gift from the ocean. It comes from the glowing jellyfish. In 1962, Shimomura discovered the fluorescent protein in jellyfish, so he won the 2008 Nobel Prize with Tsien. But at that time, Shimomura did not realize that fluorescent protein had become an important tool to reveal the meaning of life. Tsien was looking for gene proofs that could reveal genetic compilation that is different from traditional chemical fluorescent dyes. Previous studies have suggested that fluorescent protein emit light because of a reaction between certain amino acids to form chloroforms. But Tsien found that the only assistant to this luminescence reaction was oxygen. So he concluded that GFP gene could be expressed in any organism because oxygen existed everywhere. However, the fluorescence of GFP at that time was not stable and the light effect was very weak. It took a long time of ultraviolet radiation to be used for experimental observation. So Tsien's team selected a brighter and more stable green fluorescent protein by selecting mutation from a large number of fluorofiltration. he succeeded in deriving a series of colorful fluorescent proteins from a green fluorescent protein. He gave these colors a unique name, such as honey yellow, banana yellow, cherry red, grape purple, red plum purple, and so on. GFP have become a fundamental fixation in life science labs around the world, allowing researchers to look into cells or whole animals to watch the whole molecules intact in real time and ask questions once thought impossible. Our work is often described as building and training molecular spice. Tsien once said, molecules that will enter a cell or organism and report back to us what the conditions are, what's going on under the biochemistry while the cell is still alive. Winning the Nobel Prize could not stop his work moving forward. He wanted to apply his research to clinical work. Working with colleagues, Tsien helped develop injectable fluorescent peptides, as we can see from the picture here. It caused hard-to-see peripheral nerves to grow, and it allowed surgeons to avoid them to removing damaged or cancerous tissues. Tsien investigated visualized cancer in other ways or may be treated. He and his colleagues have designed U-shaped peptides able to carry other image molecules or chemical therapy drugs to target cancer cells. His lab created a new generation of fast-acting fluorescent dyes. Its optical highlights electronic activity in neural membranes, reviewing how brain cell function and interacts. And using a modified plant protein, they create the new type of genetic tag visible under an electron microscope, allowing researchers to see life in unexpected detail. Although he has passed away 3 years before, he left so much to the world. As his wife Wendy said, he was ahead of us all. He was ever the adventurer, the pathfinder, the free and soaring spirit. Courage, determination, creativity, and thoughtfulness were hallmarks of his character. He accomplished much. He will not forgotten. This is today's Nobel Prize winner. Thank you for watching.
大家好，对于自己的攻击，钱永健是这样打趣的：我只是把一本晦涩难懂的小说，变成了一部通俗易懂的电影而已。美国圣地亚哥大学的主任马克·爱丽丝曾经这样说过：他拥有世界上最美丽的大脑，不仅因为他能深入思考科学的空白领域，更因为他擅长发现新问题，并提供新的研究工具，以此帮助更多的科学家一起来研究新问题。是的，他的职业生涯一直在致力于发明新的工具，并慷慨地馈赠给其他科学家。2016年8月24日，这个让色彩进入细胞内的天才科学家钱永健逝世，享年64岁。对于钱永健的逝世，大家都为科学界失去一位聪明的天才而惋惜。钱永健的妻子更加肯定的是他性格中的魅力，他更像一名寻路人和冒险家，勇敢、果断，充满创意与智慧。他打着灯，在科学之路的前头，带着后人不断探索前行。钱永健将永远被怀念，被他的家人、朋友、同事以及很多其他因他的出色发明而受益的科学家。 好了，本次课到此结束，谢谢大家，再见。